The overall goal of this procedure is to improve the sensitivity of flow cytometry in order to quantify the specific immune cells in the injured spinal cord tissue via the removal of lipid myelin debris using an OptiPrep gradient system. This is accomplished by first dissociating the spinal cord tissue using both mechanical and enzymatic means. Then, 4 OptiPrep gradient solutions are created and used to separate lipid myelin debris from cells like neurons, glia, white and red blood cells. The cells isolated from the injured spinal cord tissue are then immunolabeled for specific immune cells like PMN neutrophils, ED1 macrophages microglia, and CD3 T cells. These immunolabeled positive cells are then quantitatively assessed by flow cytometry. Ultimately, results show the removal of lipid myelin debris enhances the quantitative detection of cellular inflammation in the injured spinal cord tissue using flow cytometric analysis.
The main advantage of this technique over the traditional cell dissociation method is that you can effectively separate and remove myelin debris from cells and thereby facilitate the quantitative assessment of cellular inflammation in the injured spinal cord tissue using flow cytometric analysis.
Begin the dissociation of the spinal cord tissue by putting Hanks buffered saline solution, or HBSS, in a petri dish. Then, using fine scissors, mechanically dissociate spinal cord segments T8 through T10 of uninjured control or contusion-injured spinal cord tissue at T9. Dissociation of an uninjured spinal cord is shown here, but the dissociation of the contusion-injured spinal cord can be done in parallel. Centrifuge the tissue bits in a 50 milliliter tube for 1 minute at 2000 rpm at room temperature.